Hi, my name is Judy, and I'm a rising senior at Johns Hopkins University, and I am studying biomedical engineering. This summer, I had the opportunity to participate in the NIH SenNet CUSP internship program with the Lin Lab at the Harvard MIT location. This summer, I contributed to the project Multi-modal Spatial Multiomic Biochemical and Vibrational Landscape of Cellular Senescence at Molecular and Chemical Resolution. To give an overview, senescent cells are poorly understood because we lack a comprehensive view of their genetic, biochemical, and cellular diversity. To address this, we created SenNetRaman Omics, a new platform that combines high-throughput single-cell and spatial multi-omic profiling with label-free Raman microscopy and machine learning. This allows us to map both the genetic and biochemical landscapes of senescence in mouth tissue across different ages. By integrating multimodal data, we identify new transcriptional signatures, gene pathways, and chemical shifts like lipid, protein, and nucleic acid associated with senescence. The result is the first multimodal spatial atlas of cellular senescence, which links gene expressions to biochemical changes across tissue and ages. This atlas provides a valuable resources for future studies on aging and cellular senescence, while the platform itself can be applied to study other cell states and systems. We use three main techniques for our multimodal mapping. The first is chemical imaging, specifically Raman microscopy. Raman imaging is Raman scattering, which is a phenomenon that occurs when light interacts with the molecule in a material. When we shine a laser onto a sample, most of the light is scattered in the same way it came in, which is called Rayleigh scattering. However, a small portion of the light is scattered to it at a different wavelength, which is what we call Raman scattering. In the image, you can see how this process works. When the laser hits a sample, some light scatter off without energy changes, but shown in green. But some of the light lose energy or gain energy by interacting with the molecular vibration of the material. And this scattered light shift in the wavelength is what we call Raman scattering, which is represented by the red line vibration in the sample. Now, shown on the graph on the right is what the Raman spectrum looks like. It is a plot of the intensity of the scattering light as a function of this energy shift called the Raman shift. Different molecules produce a unique pattern in this spectrum, like a fingerprint. So by analyzing this spectrum, we can identify the chemical makeup of a sample. What makes Raman microscopy powerful is that we can scan across a sample in 3D, collecting Raman spectrum at each point. This allows us to create a detailed image of the sample's chemical composition, which is shown in the colorful image on the top right. Next, we use StarMap for spatial transcriptomic technique. StarMap stands for Spatially Resolved Transcript Amplicon Readout Mapping, and it is a cutting-edge technique that we use to map gene expression in 3D at single-cell resolution, especially in complex tissues like the lung and the skin. First, we start by embedding the tissue in a hydrogel, and this is important because it preserves the RNA molecule inside the cells and keeps everything in its original position for sequencing. Then we use the snail probes, and these probes specifically bind to the RNA molecules we're interested in. And once the probes are attached, they amplify the signal by creating what we call DNA nanobots or amplicons. And this amplification step ensures that we can detect even the tiniest amount of RNA with great accuracy. Then we come to the sequencing part, and StarMap uses a special method called SIDL, which stands for Sequencing with Error Reduction by Dynamic Annealing and Ligation. It's a fancy way of saying that we're reading the gene sequencing inside the tissue, but we're doing it in a way that reduces error and background noise. And here you can see an image of our StarMap old skin sample. So what's really powerful about StarMap is that we can simultaneously target and barcode hundreds or even thousands of genes. And after sequencing, we can capture everything through high-resolution imaging, which gives us detailed 3D images where each gene is being expressed in the tissue. For our project, we did 6 rounds of imaging and 5 channels. To break it down, we ran multiple rounds of imaging, and at each round, it helped us reveal a different part of the gene code. So if the gene is represented by a series of number like 1, 2, 3, the first round of imaging will identify the first digit 1, and the second round will uncover the next digit 2, and the third round will fill in the more details. And by the end, we can piece together all of the full code of the gene by layering all the different rounds of imaging. And this helps us decode the gene, but also pinpoint the exact location where it is within the tissue. For our third technique, we used single nuclei RNA sequencing to analyze gene expression from mouth, lung, and skin tissues. First, we isolate the nuclei from these tissues, and they're embedded in OCT and sliced into thin sections. And we then load it into the 10x Genomics Chromium controller, which prepares the nuclei for sequencing. And for library preparation and sequencing, we use the Chromium Next GM single cell reagent kit to prepare the RNA expression library. And we loaded about 8,000 nuclei per sample. And these libraries were then sequenced using Illumina Next 550 sequencer. The raw sequencing data was then processed using Cell Ranger to generate count matrices, which were further processed in R using SoupX package to remove ambient RNA contamination. Then for downstream analysis, we did UMAP and other expression analysis to help us understand and shed light on the biological differences between these tissues. This is an overview of our workflow. We started by collecting lung and tissue samples from P16M3MR mice, which are commonly used to study cellular senescence. These samples were cryopreserved in OTC blocks to maintain their structure and molecular integrity for further analysis. The first step in the analysis was sectioning the tissue for two complementary techniques, Raman imaging, StarMap, and single nucleus RNA sequencing. Raman imaging is a label-free technique, which means we didn't need to introduce any dyes or label. This method allowed us to capture the biochemical data based on how the different molecules in the tissue scatter light. And with Raman, we were able to obtain detailed molecular composition data from the tissue. And once Raman imaging was completed, we proceeded with StarMap on the same tissue sample. Since Raman is label-free, the tissue is still intact and suitable for StarMap. And using StarMap, we mapped the expression of over 1,000 genes within the same region of the tissue, giving us high-resolution spatial transcriptomic data. And this allows us to connect gene expression with the biochemical profile obtained earlier from Raman imaging. And to merge these different data layers, we align the Raman and StarMap data set. This alignment enables us to map the gene activity directly onto the biochemical features identified by Raman imaging, creating a detailed molecular and spatial map. Next, we apply imputation to integrate the snRNA-seq data into these spatial profiles. By doing this, we could link gene expression from individual cells to their exact location onto the tissue, providing a rich multi-modal view of both molecular and spatial information. And finally, through the multi-modal integration of transcriptomic and Raman feature, we can then discover new senescent signatures. And these signatures represent unique molecular patterns associated with gene and senescence. Aging and senescence, which offers valuable insights into how these processes affect tissue on a cellular level. And to go into some preliminary findings, here I am showing the spatial mapping of cellular senescence in aged and young lung and skin sample. We conducted a spatial analysis of the cell type in both young and old lungs and skin tissues. And the goal was to map out the distribution of various cell types and observe potential differences in senescent cells between age and young and aged tissue. In figure 4, you can see spatial maps that show the localization of different cell types in lung and skin tissues. These maps were created using spatial transcriptomic and label transfer from single nucleus RNA sequencing clusters done earlier. For the lung tissue, we identify 16 cell types, including AT1 and AT2 cells, fibroblasts, macrophage, endothelial cells, and several immune cells like T cell and dendritic cells. Despite the presence of senescent cells expressing markers like P16, P21, and SEM-Mayo, there were no clear difference in spatial patterns between young and old lung samples. Similarly, in the skin tissues, we mapped 20 cell types, including basal keratinocytes, fibroblasts, hair follicle stem cells, and macrophages. And once again, senescent markers were present, but no significant spatial pattern differences were found. And for the cellular composition of the lung and skin sample, Figure 5 shows the stacked bar plot that shows the composition, and in panel A, the bar plot shows the major cell type in the lung tissues. And the most prominent populations are the AT2 cells, macrophage fibroblasts, endothelial cells. And these plots allow us to compare the proportion of different cell types between the young and old lungs. While some are different in proportion, the overall cellular makeup remained consistent across age groups. And in panel B, it shows us the cell composition in skin tissues, which again, no major shift in the distribution of cell types. This analysis provides insights into the presence of senescent cell in both young and old tissue, but without significant spatial pattern differences between the age group. And this helps us understand how aging impact different cell population in lung and skin tissue, and also help us set a foundation before our analysis. Here, we're showing the multi-modal integration of star maps, spatial gene expression, and Raman imaging, and it shows our alignment step in our technique. So starting with the first row, we can see the different image modalities. On the far left is the star map DAPI imaging, which highlights the nuclei of the cell in the tissue. This serves as a reference for cell locations. The next image shows registered star map DAPI, which aligns the DAPI signal from star map with the RNA, DNA data. And the Raman DNA image displays the biochemical information specific to DNA, capturing using Raman imaging without the need for fluorescent labeling. And you can also see the registered star map DAPI and Raman DNA image, which combines the gene expression and biochemical data, showing how the two data set overlaps. In second row, we see a lot of the images with cell segmentation, and you can see the star map segmentation where individual cells have been identified and outlined based on star map data. And the second image registers star map segmentation with Raman data, which aligns the segmented cells from star map with the Raman imaging data, emerging gene expression, and biochemical information. Finally, the segmented Raman DNA image also shows the cell boundary purely based on Raman imaging, providing additional biochemical insight. Here, we just wanted to show how the overlapping, alignment, and cell segmentation comes into play with our multi-modal platform. As we move forward, we're continuing with downstream analysis of the integrated star map and Raman dataset. This multimodal approach is providing us with new layer of information, revealing subtle patterns in cellular behavior and gene expression that were previously difficult to capture. Our current focus is identifying new cellular senescent features by combining biochemical signatures with spatial transcriptomic data. With this approach, we're able to pinpoint exactly where senescent cells are located in the tissue and how their gene expression correlates with their molecular environment. By integrating these data sets, we can uncover previously unknown senescent markers and cellular interaction that could play a key role in aging and disease progression. Thank you for watching, and I wanted to extend my gratitude to the NIH SenNet CUSP internship program this summer for the amazing opportunity that I had at the Zhou Lab at Harvard. And I also wanted to thank my PI Jian Shu and all the members of the lab for such a collaborative environment, and I really learned a lot from everyone, and everyone was so helpful. This experience was really transformative for me because it helped solidify my passion in research and STEM, and it also helped me find my true interest in studying senescence and aging in the future as well. Thank you so much.